All right, I think it's 10 o'clock. Good morning, everyone. I really didn't expect this kind of audience, so this is kind of unnerving, I would say. But yeah, because there's such good, amazing talks just happening at the same time, so I was expecting maybe like 10 people would just show up for this, and okay, I hope I can entertain you for the next 40 minutes. So what are we going to do in the next 40 minutes? I'm trying to clarify what personalized medicine is, refresh your high school biology. That's going to be a little boring, but just bear with me a little bit because I do want to explain how the genomic data actually is turned to genomic data from like the DNA, right? I mean, we literally have something physical. So how do we put that into computer programming or just any type of analysis? Like, how do they look like? Are they numbers or whatever, right? So those are the questions we will try to answer. And yeah, I will also show just a little bit how this data is analyzed, and then potentially talk a little bit more about why we care about cloud computing when it comes to especially genomic data, and why Google Genomics, for example, is investing a lot of money for that kind of stuff, and why do they care. And if we have time, we'll also talk about some ethical dilemmas, if you will. Yeah. So what is personalized medicine? It's also termed precision medicine. I use personalized medicine just because it's a sexier name, I'm not going to lie, just to attract more people. I guess I kind of achieved that. But people like to use the precision medicine because personalized medicine kind of sounds like we're making a drug just for you. Oh, well, that's not the case. We're actually just analyzing the data coming from you. It could be all sorts of data, by the way. It doesn't also need to be just your genomic data, right? It could be all sorts of sampling data, and I will just give a couple of examples of that. But it can be just about, what your lifestyle, or how many times have you been hospitalized? Do you have like some other chronic diseases? So we just collect that and then try to figure out what kind of treatments we should give to you, right? So that's the idea of personalized medicine. Then not necessarily that we're making new drugs for you, although that's actually kind of happening a little bit too with new immunotherapies nowadays in a way. But okay, I'm not going to get into that today. And I'm also going to admit just right now, so when I submitted this talk, I didn't realize it's only 40 minutes, dude. Like, you have to be aware of what you're talking, right? So, but I will try to be quick and at least get a little bit taste of what we're doing. So, and then what is pharmacogenomics then? That's actually another big term that is used a lot with personalized medicine or precision medicine. And according to NIH, so the National Institute of Health, pharmacogenomics is the study of how genes affect a person's response to drugs. Pretty simple idea, right? And it just combines the genetics and pharmacology knowledge, and then we basically try to figure out then what kind of dose we should give you, or we should actually give you this drug at all or not. So the idea is, again, pretty simple. We have a population of patients. They have the same condition. They have certain disease. And then we collect samples from them, right? And we do some sort of DNA profiling, and then we group people into good response, no response, or bad side effects. And here, actually, where the genomic data science is applied, right? You can imagine that, okay, now we have this data. How do we actually group these people? And it's actually pretty complicated, so I will not be able to touch on everything. But you can also imagine a little bit now, right, how the machine learning algorithms potentially might be applied here. So we will actually want to be able to do some sort of a classification, clustering, etc., right? But precision medicine isn't also just about pharmacogenomics. So it could also be just people without symptoms. And we do some sort of DNA profiling and apply genomic data science again. And then we decide what kind of people or what group of people have high risk of cancer, what type of cancer, right? And or some of them with maybe with the lower cancer risk. And it's again the same thing. We can also have a population who already has the disease, and we might specifically want to look what's actually happening in that tissue, because cancer is a pretty complicated disease. And although we just say cancer for everything, it's actually not true. Even if when you have colon cancer, it's usually there are a bunch of subgroups that we need to figure out so that we can give you the right treatment. So, again, for example, we can have the patients with colon cancer. We can do DNA profiling or RNA profiling, and I will mention that in a second, of the cancer cells. So before, the sampling could be like, I don't know, saliva or just blood. So we would just look for your DNA, right? So it's basically all the same all across your body. But when we're specifically looking for the tissue, things actually can be a little bit different than what you were born with, right? That's the actually main problem with the cancer, is that the genes aren't the same anymore. You have a bunch of accumulated mutations. The cells are just going crazy. They're totally rogue. They're really fighting against your body, and unfortunately your immune system is not recognizing them enough, and they're just not fighting against them, right? So here, again, we apply the genomic data science, and then we can just find the subtypes of this cancer and potentially give the right treatment to the patients. So what is genomic data science? I know you've probably seen this a lot about, from the data science as well, right? It's not a new thing. So, but I mean, as you can imagine, it's the overlap between computer science, statistics, and biology. And again, the scientists, they just love coming up with new terms. Every new word is more sexy, and then, there's a specific reason for why they call it, and they never want to give up their term. So we don't just call this, obviously, genomic data science. Although I'm going to admit, again, I think this sounds more sexy, and I think it's a little bit more clear than bioinformatics, for example. But they're kind of the same, you can imagine. Or computational biology. So some people also have really specific definitions for these fields as well, where, for example, bioinformatics is usually the field that produces the software or the code, basically, that the computational biologists then use to figure out what the biological processes are. But I think, basically, the idea is kind of the same, okay? So you use the domain knowledge, and you do some stuff with it, right? And then you figure out your groups and potentially what's happening within your data. And that's actually data science, basically, at the end of the day. So the only thing that we're changing here, instead of the domain knowledge, our domain knowledge is now biology, right? Something really specific. So, a little bit of high school biology refresher. Here we have our DNA, right? You guys probably remember this. So the building blocks of the DNA are the bases, right? We have 4 of them. So A, T, G, and C. And A always is paired with a T, and G is always paired with a C, okay? So we have 2 strands, and both of the strands are complementary, right? So things get a little tricky because we do have 2 strands, but we also have 2 chromosomes, or 2 DNA, let's say, all right? One from the mom, obviously, right? And then the other one from the dad. And we have 22 chromosomes plus the 6 chromosomes. So basically the size of the human genome, actually, when we look at it, right, we have 3 billion bases, so these guys right here, and times 2, because we're getting some from the mom and the other half from the dad, we're ending up with 6 billion. And then we have 22 chromosomes, which makes obviously 44, and then we either have the XY or XX, right, making it 46. So it's kind of hence like the 23andMe, right? Just, yeah. All right. So again, this is our DNA. And then if you remember, from the DNA, we actually produce the RNA. So the DNA is the big guy of the cell, right? It's the brains of the cell. And it always remains in the nucleus, if you remember just a little bit. It doesn't necessarily gonna leave it, right? It's the big guy. It's a huge thing, like literally. It's just 6 billion bases. It's really not a small thing, right? So it needs some sort of a messenger that will actually take that information from the DNA and potentially go out and produce some proteins. So that guy is actually the RNA. And we say that this is a transcription. And what happens is, again, we're using these bases, right, to move along or give the information again outside. So it's basically the same idea. We have this recipe, now we're going to apply it, right? And after the RNA, we turn this into a polypeptide chain, which is basically the protein. And what proteins are is actually what they, what you are. They're literally the building blocks of your whole body, right? So basically, what's happening in your DNA actually defines what kind of proteins you will have, potentially how many of those proteins you will have, right? And that's also connected to the RNA. So this axis here can be different in a couple of different ways. So either the sequence can be different, and then it will actually be passed along, right? And then we can potentially have a different structure of the protein. Or we also have something called gene expression. So basically the amount of this transcription, how much of the mRNA will be floating around the cell so that we make so much of that protein, right? And that's, you can think, is again more of a numeric data then, instead of like a discrete one, right? So it defines how much of that protein will be actually allowed in the cell. And this is basically the central dogma. So you guys remember this, right? And also another important thing is that our DNA, okay, is huge, but it's a really small percentage is actually a gene. And the description or the definition of the gene has been changing a lot, especially lately, ever since like the Genome Project, and even nowadays, everyone has just different ideas. But we can still say that a gene is basically the part of the DNA where we can generate an RNA, all right? And then which translates into a protein, all right? Stop me if you have any questions. This is clear though, right? So far, nothing weird. All right, and then so obviously from the proteins we can also go to the metabolites, and that what whole biological processes happen. And of course, metabolites are also connected to your environment as well, right? This is where the environment also comes in terms of the molecular biology, and this is what gives us the phenotype, or this can be basically the phenotype can be just anything, right? You can be your height, it can be just color of your eyes, or it can be your disease, okay? And this is what we're interested in. So we're trying to understand what's happening along this axis that gives us this output, all right? So just to give you a little bit of an idea, what kind of data then we have, right? So we already talked about the DNA, and what we call it, the genomic data, basically, right? You can actually have two types. So one would be the germline, which is what you're born with, right? So that's going to be all over your body, all of your cell will have that same information. But when it comes to somatic, like I talked with the cancer, you can accumulate a bunch of different mutations, and it's going to be specific to that tissue. And in that case, you can imagine you have to do different analyses because it's not going to be the same. So obviously, like if you just collect the saliva, right, it's going to give you more of the germline DNA, and it's not necessarily going to give you the DNA from your tumor. So those are two different things. And again, what type of data we're talking about with this? It could be discrete and continuous. And I will just show you in a second how we turn it into a continuous data. But discrete, you can kind of imagine, right? We have sequence differences. We try to determine those sequence differences and potentially do some analysis with them. And then when it comes to RNA, again, we call it the transcriptomic data. And again, this is going to be very specific to the tissue and cell type, actually, because that's what defines your liver cells from your eye cells, right? They're totally different, but they have the same DNA. I mean, as long as you don't have cancer, let's assume you don't, they're going to have the same DNA because they're having exactly the same germline DNA, right? What makes a difference is the expression of this RNA. So the level of the proteins, certain regions of the DNA actually being open to turn into protein, and that's what makes the difference between the tissues and all of the cells. So it's going to be entirely different, and it's going to be very specific to tissue, cell line, and it's going to change within the hours. So for example, if we have cells, treat them something, right? You have to wait about a couple of hours to really see the difference between the gene expression, because it's going to be, the cell will give some sort of a response in terms of the mRNA, right? And that's going to happen, it's going to take a couple of hours. Then we have the proteomic data, which means that at a specific time in a specific cell, we're looking all of the available proteins. And it can also be discrete and continuous because it's, again, depends on the question you're asking. if you want to just see what proteins are actually available at that time point in that specific cell group, or you can just ask the question, how much of these proteins are available in a certain time point in the cell group, right? And this is going to be minutes. Response is just like, now we're actually going in minutes, right? And when it comes to metabolites, it's basically the similar idea. Again, we can just ask the question, what kind of metabolites are actually available in the cell versus how much of them is actually available, right? And this is going to actually take seconds, so everything happens like incredibly quickly. So today, I'll be mainly talking about the DNA and, well, I'm not even going to, unfortunately, get into the RNA, actually. But these two platforms are quite different. So if you have any analytical chemists in the room, you might know the mass spectrometry platforms. That's how actually they detect these stuff. So it's definitely much more like chemistry. But when it comes to the DNA and RNA, they're actually analyzed pretty similarly. Because you can turn, so we have different enzymes, we can actually turn these little pieces of RNA into DNA and get the same kind of information because we remember, right? We know that the strands are complementary. So we can get the same kind of information by turning RNA into DNA and basically do the same kind of sequencing, applying same kind of sequencing technologies or whatever, and do kind of a similar analysis. Again, the difference is potentially going to be, mostly we'll be more interested in how much RNA there is actually in the cell. Any questions so far? Yeah?
We're talking about time. I didn't know what
So how much, so for example, the response of the cell and how quickly actually these things really change in the cell. Yeah. Great question. Any other questions? All right. So yeah, when you're doing experiments with these two, you have to be careful, right? If you're really going to try to capture something, that's why it's really difficult. And these fields are still kind of emerging. They're relatively new fields, but we are actually trying to integrate all of this kind of together. So that's the systems biology, actually, if you will. Okay, so quickly let's talk about the Human Genome Project. So this was a 13-year-old project coordinated by the U.S. Department of Energy and National Institutes of Health. And the aim was to map and identify all genes of the human genome to determine the coordinates of the human genome. So when we talk about any kind of data, when it comes to DNA, we always talk with the coordinates, right? Where is this change is happening? Which chromosome, which base pair? I told it was like 6 billion, whatever, right? So 3 billion when we think in terms of the coordinates, right? We don't necessarily add another coordinate for the maternal or paternal. We just look for that same location, and I will just show you in a second. But we always think in terms of the coordinates. So that's why, actually, this Human Genome Project was so revolutionary, because it let us know to have, actually, now we have a map, okay? We still don't know a lot of things, right? But we at least have a map. We know where things are, and we can understand what the changes potentially mean. Because we know what those genes are, we kind of have an idea what they're doing, and so on and so on. So the first human is also produced the first human reference genome, and what you see here is actually printed version of the first human reference genome. So you don't even see the volume 1, which is the chromosome 1. It's probably in a whole other library. But you can't imagine, this is literally just the letters, and this is how big it is, just to give you a sense. And the new builds of a human reference genome always comes like every several years, and then it's almost kind of like giving a crazy system update for biologists. So it has also its own challenges. But again, so whenever we do some sort of an experiment, we always refer back to the reference genome, right? Because that's our map, and we're trying to figure out when we get this data from a sequencer, whatever, we're trying to find out where those changes are happening according to the reference genome. Okay, so what is these changes I've been talking about? What is genetic variation? It's basically the differences in DNA content and structure among individuals, and any two individuals, humans, have about 99.5 identical DNA. And even when it comes to chimps, we still have about 99%. So it's still quite similar. But the human genome is pretty big, right? So we're still talking about 3 billion base pairs. And of course, even that small percentage will turn into pretty huge numbers. So we will be talking about actually hundreds of millions of variants in an individual, okay? And that's what actually makes the data big, right? So we have a lot of individuals, and all of these individuals have some sort of an information for these 100 million data points. Okay, you get the feeling? All right, so the types of genetic variation. One is a single nucleotide polymorphism. You don't need to remember, whatever, just the name, right? It's just a SNP. That's what we call them, okay? SNP. So it's just one base change. It's only one base. Very simple, right? Another weird idea though. And we can basically say that it's just like a DNA spelling difference. And then we have much bigger changes, which are then insertions and deletions. And then we have even bigger changes, like huge chunks of DNA being different among individuals. So this one is a more difficult one to actually analyze. And you definitely need the sequencing data to analyze this kind of stuff. But this one is very popular because we actually know the structure of this in populations, especially in European populations. But most of the DNA kits and stuff are actually looking for SNPs. So for example, 23andMe, the Ancestry, Gene by Gene, etc. So all of these guys, usually when you give them your saliva, they will be looking for these SNPs among your DNA because they're easy to analyze and they're quite frequent actually. We do see a lot of differences in the population, so we can make more sense out of it. So let's talk a bit more about this. We already know it's just the variation in single DNA base pair, blah, blah, blah. But it has to have an allele frequency of 1%. So what is this allele frequency? And what is an allele here? So you probably remember a little bit about the Mendel and the P's, the mother and the capital A and the small a. Well, it's a very similar idea. So as we mentioned before, we have one chromosome from the mom. We have another one from the dad, right? And just to be clear, we have a DNA strand, right? That's also 2. So I know it's kind of confusing. So you have one dad, it's just like too many 2s. But just to be clear, right? So you have one strand from the mom, and you have another strand from the dad. And then we see this difference right at the sequence, right? So the rest of it is actually exactly the same. And what I can do, because the information is actually just repeated, right? So there is no difference between these strands. They actually give me exactly the same information. So what I can do is I can just take one strand as a reference. Let's say that's the plus strand. And then get rid of the other one. And yeah, let's say this is also the specific location on chromosome 2. We already saw those were the chromosome 2s. Oh, by the way, chromosome 2 is an interesting one because that's the one that we have like this huge difference between the chimps. All of the apes actually have 48 chromosomes and humans have 46. And it's because actually their chromosome 2 is split into 2. But in humans, somehow they just kind of merged together and then made one chromosome. But yeah, like I said, it was a weird side story. We can just keep one strand, right? And keep talking about this reference strand all the time. Because otherwise we just need more and more space and data to actually collect that information and keep it. All right. So then we talk about the genotype. So now I think it's easier to see, right? So we have 2 alleles right now. What the allele is, what makes up this genotype, all right? So basically T is one allele, right? So when you think about the P's, the capital A, small a, exactly the same. So one is the T, and then the other one is the C, giving us the genotype for this orange person, who is not the President of the United States, but it's just an orange person, okay? Okay, so then we have also a turquoise person here at the same location, not perfectly aligned, but they're the same location. And then we can imagine this person is going to have what genotype? Yes. And then the purple guy is going to have, yep, perfect. So How are we going to turn this into some sort of a numeric or some continuous variable? We're actually going to look at the dosage of one of the alleles. So we'll pick, again, exactly the same way we did with a strand. We'll pick one of the alleles, and we will count how many of that person actually has. It's a pretty simple idea, right? So if you think that we're counting the T's, what's the dosage is going to be for the orange person?
1.
Yep. And what's going to be for the turquoise guy?
2.
Yep. And then the guy, the
0.
purple? 0. Exactly. It's very basic, right? And now we actually have a continuous variable. We can put in any kind of model. You can do any kind of analysis that you want to do. It's a very simple idea. And usually what we do is that the thing that we usually count is the one that is less frequent in the population, because we expect to see less frequent things to actually affect you and cause the disease, because we have something called natural selection, right? So we don't expect the bad things to really be more frequent, right? We expect bad things to be less frequent. And we want to count, or we want to see how much that bad allele is actually affecting the disease. So we can get a gradual kind of a measurement, if you will, right, with that dosage and add it into your model. It's actually pretty simple. So for every increase of that one allele, you will see this change in the phenotype. It's a very basic idea, right? Okay, and then we also have something called haplotype. So I know sometimes, like, when we talk about this really arbitrary stuff, we forget that they're actually physical things. Like, this is literally molecules in your body, and they have to be inherited, right? Passed along from your dad, from your mom. And they don't change and get scrambled that much. So what happens is, a lot of the SNPs that we just talked about, those little singular things, right? They happen to actually be inherited together. So, and they happen to be inherited like together in chunks, actually. Because if you remember, again, a little bit from high school biology, there's something called crossover. You remember, like, the chromosomes were getting, like, exchange and whatever. That happens not very frequently. And even when it happens in really huge chunks, right? So things still tend to remain together. And when you look at specific populations, like the European population, we have really long haplotypes where a lot of SNPs, remain together because, well, I guess European populations have been through a lot of bottlenecks, a lot of death, plague and stuff. So they had to found the whole population from a really small group of people. So they didn't have enough time to scramble around with the whole crossover and everything to have these smaller haplotypes, like, for example, the African populations have. They have much shorter haplotypes, so they have much more scrambled, actually. They have much more diversity within the population compared to Europeans who just like white nose, right? Okay, so when we get to our haplotypes, again, here we see the two strands, right? So let's be clear again. This one is from the mom, and this one is from the dad. So again, we're just looking for that plus strand, right? And then here again, we have the haplotypes. Oh, my orange person haplotype turned into green, so sorry about that, but that's supposed to be orange. And then again, I can apply the same idea, right? I can always have the reference alleles, and I'm just counting them again. And then these are the dosages I will then have for my haplotypes. So the tricky part here is I know that this one is from the mom, this one is from the dad, and so on and so on. So what's tricky about this is that, and I will come to that in genome imputation, is actually figuring this out. But we'll come to that a little bit later. This is just like an introduction. Let's try to remember this, okay? So we have before we had a distribution from 0 to 2, right? And now we only have 0 or a 1. All right, so how do we know actually all this DNA structure, if you will? We know that from the global reference genetic variation studies. So there was a huge one called HapMap. This is one of the first ones. And then the 1,000 Genome Projects. So they literally went and collected DNA samples from a bunch of different populations, sequenced them, and tried to figure out what their haplotypes are. So we can have an idea how these SNPs actually tend to be inherited together, right? And then we can potentially apply that information later on, but also let us understand how this whole heritage thing actually is happening, right? How do we actually pass along the genes and the genetic information? And it also gives us an idea how much variation we have, and all of this can be actually collected in a database. And this is how we also annotate our data then later on, right? We know that this specific change in this specific location actually means this RS987, which is already known, and you can link that to many other papers, and you can actually understand what's going on already. So we actually tag all of them already, or majority of it, let's say, especially for the SNPs, so that we can actually trace it back to what was happening with that specific variation. And biology, I think, is one of the fields that has the most databases. There are just so many databases in biology, and I'm not going to get into that today, but you can imagine for each of the axes, like when you remember the axes with the DNA and RNA, blah, blah, blah. So we have databases for genes, right? We have databases for just the variation itself. We have databases for proteins and metabolites and actually the pathways, because, you know, all of these things basically act together, and then all those interactions are actually in a database too. So it's pretty crazy, yeah. But so how do we actually identify this genetic variation? So right now there are two main ways. You can either go for a SNP array, right? So you can either use a chip, which means that you're already using available information. You're looking for these specific SNPs that was already in the database, right? And then you just measure basically what the people actually have in those specific locations. So you just look for their genotype. Or you can do next generation sequencing, which means you can find a bunch of things, because you are basically getting the full information about the DNA of that specific person, right? So what these chips usually have, they have about 750K SNPs. So this is how many SNPs they're actually genotyping for one person each time, right? And when you give your DNA, for example, to 23andMe, they use this kind of stuff. Although lately they also started creating their own chips, because I will show you in a second, they can do genome imputation, and they can actually yield up to 30 million SNPs by using this information that we gathered from these previous projects and the DNA structure, right? So because things are not entirely independent, so we can predict what those missing things actually are, right? And of course, these stuff are much cheaper than the sequencing as well, but it cannot detect all types of variation. So if you have like a de novo mutation, so that's something you inherited from your parents, but they actually didn't have it themselves, which means that they either had a mutation in their egg or sperm, and you have it. So, ta-da! You can only find that through the sequencing, right? Or even really rare variants that we potentially have, or the structural variants I just talked about, like big chunks of DNA being different among individuals. You can only find that stuff with sequencing. But it's much cheaper. And academia is cheap. I mean, poor guys, they're really not getting any love, especially right now from the government. It's tough. So, of course, people usually go for this for now. And also, it just gives you a very relatively easier data to work with, because you already know what variations you're looking for, right? You already know what their locations are. They're already tagged. You already know their IDs, where they're happening, etc. With sequencing, things get a little bit more complicated. And it is frequently used for genome-wide association studies, which I will come to in a second too. So when it comes to sequencing, I already mentioned most of the stuff, but it does not depend on the prior knowledge, right? So we are basically sequencing the genome of that person entirely, so we don't really depend on anything. The problem is, the data is really difficult. It's huge. It's 6 billion bytes, and even more information about them we will get to in a second. But the data is huge, so you definitely have a lot of storage problems, etc. And it's also way more expensive than actually doing chip. So it's less desirable usually for really large studies if you're not necessarily creating to use that information to create like a database, the ones I showed before. Those were, for example, usually sequence data, right? Because we want to understand what the structure is. So this is the first time we're kind of gathering that information to create a newer map, a different layer on our map. So that's why we actually need the sequencing. But if you just want to do kind of like, we just want to understand which of these SNPs really affect the cancer prevalence or something like that, you don't potentially need this much. as much. All right, so let's talk about the chips for a second. I just wanted to give you a little bit of an idea how the data looks like, or potentially even the files look like, because in biology, unfortunately, it's kind of a newer field, and people don't necessarily have a consensus. They're all different sorts of data types, and you have to have all different terminal softwares that you need to use to potentially convert one from another, blah, blah, blah. But the general idea is usually you have 3 output files. So for example, okay, these are called BIM files, whatever. They usually have the information on the variants, like I said, like the position, which chromosome on it, right, the base pair, the ID of the variation, etc., etc. And it's usually human readable. It's not that big, so you can imagine you have like 4 fields, 750,000 rows, right? It's not that huge, just 25 megabytes. And it looks kind of something like this, if you're just wondering, So we have the chromosome, the ID, genetic distance is just ignored nowadays. No one cares about this, so you can ignore too. And then the physical position in the chromosome. And then the alleles, right? Those are the things that we wonder about. And then we also have the fem file, so which basically specifies what kind of samples you have, right? If the person is like a female or male, and it's also human readable. It's usually a small file as well. It depends on the size of your sample size, etc. And then we also have the bed files, which are like binary files, which actually contains the data, the actual measurement from those genotypes, right? So it has kind of a similar encoding. So it's already defined what it is in the BIM file, right? What those alleles are. And then you can just see, again, have the similar 0, 1, 2 kind of a, basically representation to collect that data. And these files are a little bit bigger. They're like 2 gigabytes. And that's why it's 2 gigabytes. So even though they're not entirely still that small, because this is still a binary file. I mean, when you extract this, it still gets much bigger. It gets even bigger when you do the imputation. So let's talk about the imputation. Okay, you need to bear with me a little bit. This is a little difficult, but I really want to give you a little bit of sense of what we do, really, what we do, right? So here we start with what we measure. All right. So we have that distribution with 1 or 2 or 0 or 2, right? It's again, each of these rows are a different individual, and each of the columns are basically different SNPs, right? So for example, when you think about this person, we're looking at the TT, right? T being our reference allele. This person has a TT genotype at this location. And then this person potentially is a different SNP, so they can have actually different alleles, right? And potentially here, that person has an AA genotype. And the reference then we were looking for was C. Make sense? Because if the person was AC for the specific SNP, right, that would be a 1. If it was CC, it would be a 2. But this person's AA, and I know that, so it's 0. And this information is contained in the other file that I already know, right? What those alleles are, etc. But so this is how it looks like. So the problem of going from this is the genotype data, is going to the haplotype, the one I showed you before. Because that's how we're actually going to predict what the other SNPs are, right? That's how we know that they're inherited. But we cannot do that with the genotype data because it's already summed up. So we have to somehow come up with a way to split it back up with the probabilities. So what we do is we use the reference haplotypes. Again, all those huge projects, right? They already sequenced these people. They figured out their parents. They actually know what haplotypes those people have. So what was inherited as that one string of SNPs, right? And what we see here, each line is a different haplotype, okay? So not an individual, because 2 of these would make 1 individual, right? You following? Are you with me? And then the columns, again, the same. So my columns are, again, my SNPs. Make sense? So now I need to use this information to figure out what these chunks are actually going to be. Because once you can figure this out, it's easy to fill out the missing values, right? So I actually probably didn't talk about that, but all the gray stuff we did not genotype, right? Those are the things that we're trying to impute, we're trying to predict here. But you can imagine, so once we figure out actually what this chunk is, because of the structure we already know, we can just get the probabilities with some Bayesian statistics, whatever you will, and then actually predict what those values are. Right? It's clear. So this phasing part is the more difficult part, and it's computationally incredibly intensive. Right? And you can imagine, and it uses a bunch of hidden Markov models to really actually, and if you're familiar with them a little bit, it literally goes from this one stage to another, and oh yeah, where are we with the time? Yeah, it goes from one stage to another and tries to basically find all of these chunks in the DNA, right? And then we basically figure out what we had. So very quickly, again, we remember, for example, in this case, I'm counting the small a, right? And then this would be a 0 in our logic, right? And then this would be a 1, and this would be a 2. So what I get after the imputation are the probabilities of that person having this specific genotype. Does that make sense? So I can actually then estimate an allele dosage again by just multiplying these with their probabilities. So I just weight them by their probabilities, ta-da, and again, get like a nice continuous variable. And the distribution is going to be the same again. So I have a peak at 0, right? It's going to go down. I have another peak at 1. It's going to go down. And now I have another peak again at 2. Any questions? Good? And if you want to, you can also impute your genotype. So the 23andMe actually gives you just a small amount of information, and for free you can actually upload your data in one of those imputation servers, and you can get more information about yourself as well. Okay, I'm going to be really quick because I don't have much time left, and I still have so many slides. But how are we here? Do we like empty the room right now? Are we good? No question, nothing? Okay. I'll just keep going until they stop us. Is that okay? I hope you're not too bored. And then how do we do then this sequencing? Oh, well, God. All right. Real quick, okay? So we have the sample again. This can be blood or tissue or saliva or whatever. And then we just extract the DNA, and then we fractionate it because the DNA itself is super long, and that cannot just go into the sequencer. You need to have smaller sequences to actually figure out really what's going on. So that makes things a little bit more difficult too, right? Because now we've scrambled, we made them smaller, and everything is scrambled right now. So that goes into the sequencing, and what we get out of the sequencing is actually the probabilities, again, of each of these bases. Does that make sense? And the colors actually indicate, so throughout the sequencing, they emit some sort of a specific color, and that's measured. That's why there's these crazy colors going up and down a little bit, all right? Doesn't matter. From this, we get the reads. So each of these little chunks are basically the information from your DNA, but they're now chunked up, and we don't know where they actually are, right? And this is where comes the alignment. This is the really difficult part, and again, computationally incredibly intensive. So we have the human reference genome, and we need to map now all of these things to the human reference genome, right? And we will also have differences, so we need to be open to the error proneness of the whole situation, but we also need to find the right spots for all of these things. And again, this uses Borovillo transformation, and it's incredibly computationally intensive operation. And raw data is an oxymoron, right? That's definitely true in biology. It's like it never ends. You always have the raw data. Like until you get your p-value, even with the p-value, you still have raw data. So this is the data you actually start with from the sequencer. And per individual, you're about 100 gigabytes, okay? It's a huge thing. And this is actually in binary format. So you actually need to do the alignment, extract this, and do all this crazy mapping stuff. And again, you can imagine it's incredibly intensive computationally, right? And yeah, this is the entire workflow to basically actually figure out what the variation is in that specific individual. And obviously, you'll never do that for one individual because one individual never gives you any information, right? You need to have hundreds of them, potentially thousands of them. So things get huge very easy. Okay, and so very quickly, this is one of the analyses that we do, and that's what we do in our lab as well. So we do genome-wide association studies, and what it means is that we basically take the genetic information and plug it into, some sort of a test statistic. So it can be like a simple chi-squared, right? You can treat them as categorical variables, the zeros and ones and we got. Well, you can't do this with the imputational data though, imputed data, right? Because we have probabilities and we will have things in between. So you can either do cutoffs, whatever, but you can use very simple statistical tests. You can use very simple linear regression. You can use multiple regression. And again, one of these is going to be like your allele dosages, right? Because we have continuous data, and it's actually more easy to work with than this stupid chi-square. You can adjust anything. I mean, this is like, nah. Or you can do your craziest splines, cubic splines, or, whatever you have, right? But the problem is, you need to apply this now to 9 million SNPs that you probably got from your imputation. Does that make sense? That's a big problem, because you have a crazy multiple testing correction problem, right? So this is how the data looks like. So this is a Manhattan plot that we very frequently use, and I know this is like super bad resolution. It's from an old paper, but it was an important one on lung cancer actually. And what it is that on the x-axis we have the positions of the SNPs. So each of the dots is one SNP, and on the y-axis we have the minus log 10 p-value. Minus log 10. You are understanding this? And then this is our cutoff. So in order to actually pass our multiple test correction, right, it has to be 0.000000005, the p-value, right? Because we have to actually correct for that. And the problem is that we know not all of the SNPs are independent. Oh my God, how are we going to deal with this? It's crazy, right? So that's why we usually just correct for 1 million. Because we assume that there's at least 1 million independent SNPs. That's what goes into the chip, right? And then from the chip, we use kind of the correlation of all of those SNPs to impute the other stuff. So we can imagine that the imputed things aren't necessarily independent. Does that make sense? But it's a very hard threshold to pass, right? So to get something significant that potentially says that it's really affecting your phenotype, it's a tough one to get. But as you can see, this one is a good study. It has this really nice trace, right? So what we also know is that the SNPs, again, appear together, right? They tend to have the same kind of genotype information then. So if one of them is associated with something, then the other one that is correlated with it also going to be associated with the phenotype as well, right? It makes sense. So that's why we actually see the spike of the p-values. And when we see something, some loners like, kind of like this one, we say, it's quite likely it's a false positive because I don't necessarily see that trace. The other things are not necessarily appearing with it, right? So this would show that, for example, then definitely in this region, something is happening and, you need to look into that and that's definitely affecting your phenotype. All right, and this is a really cool one too. So one of the problems that we actually have is the population structure. So look at this map. This is literally just a simple PCA. And you can actually map it on the European map, and you can actually figure out where the people are coming from. So that's how tightly knit the population structure is with our DNA. So, and you can also imagine, if you try to analyze different populations in the same analysis, you definitely have a problem of mixed models, right? You have a problem of basically different structures being actually analyzed together, and you cannot necessarily do that. So you either have to deal with this, potentially add several of these PCA components to your model or something like that, but this is definitely a hard thing to deal with. So that's why usually right now we have a lot of European studies, right, because they're easy to collect, people trust the government, etc. But it's really difficult for minorities. Literally, they just don't give samples and there aren't enough studies, and that's actually one of the ethical dilemmas, if you will. All right, I'm gonna skip this stuff, but I'm gonna talk about this real quick too. So, and another problem that we have, I mentioned before, is that when something is very deleterious, right, it's going to be super rare. So you know that that thing, when you have that one variation, you get a specific disease. Like Huntington's, right, you can just detect, we know genetic tests, okay, I need to go. But yeah, so that's one of the problems, is that we cannot figure out the common things, how it's actually affecting the phenotype. So a lot of things we know it's actually inherited and it has a genetic component, but we cannot find the locus for that, right? And the reason is the whole genome is actually acting together. It's not necessarily acting independently singular. So this GWAS, approach that we have is actually pretty bad. Yeah, and that's why we actually try to do the network analysis and try to analyze a couple of things together, right? And this is where it comes into like kernel machine tests and network analyses. So I'm sorry, I will end this right now, and I couldn't get into cloud computing. But our lab is also looking for people, so if you're interested in any of this kind of stuff, just come find me. My name is Ezgi, and I'm happy to talk to you about any of these things, okay? Thank you very much. I hope I didn't bore you too much.